Good morning, good afternoon, good evening to everyone around the world. Welcome everyone to the parallel session portion of the Global Conference on Aquaculture Millennium Plus 20, the day 2. My name is Rohan Subasingh, and I will be your facilitator today. Just so we are sure everyone is in the right place. This is the parallel session stream 2. That's the green stream. We have 2 sessions today. The first is about aquatic genetic resources and seed supply, followed by a session on biosecurity and reducing the burden of disease. The thematic sessions were proposed by the International Program Committee for the conference, and a group of experts have drafted papers on each thematic topic. These papers identify the current status, the issues and challenges moving forward, and formulate recommendations on how these can be addressed for each thematic area. These draft papers have been posted on the GCA conference website since the beginning of this month, and the participants are invited to review the papers and provide feedback to the Secretariat, and this opportunity will remain open until 15th of October. These feedbacks will be considered by the authors in finalizing the papers for publication. The presenters today will present summaries of the key findings of these thematic review papers, and the feedback provided in the panel sessions will also be considered in finalizing the papers. Please note that this session is being interpreted. Please remember to select your preferred language of interpretation in the interpretation button at the bottom of your Zoom window. I would like to introduce our local moderator for the session today, who will be moderating the room in Shanghai for the participants who are presented there in live. Professor Chen Songling of the East China Sea Fisheries Research Institute, who you can see on your screen here. And can I say how great it is to see so many people able to participate in person. I think in Shanghai, there are many sort of participating live and listening and seeing things on screen. Professor Chen Songling is a chief scientist on aquaculture biotechnology, the Chinese Academy of Fisheries Sciences. He is also a professor at the Yellow Sea Fisheries Research Institute and a director in aquaculture biotechnology branch of the China Society of Fisheries. He is an expert in genetic resources, exploration, and molecular breeding in marine fishes, including marine fish genomics and exploitation of genomic resources. Allow me to introduce our speakers today. First, we have Dr. Anna Sonnesson from the Norwegian Institute of Food Fisheries and Aquaculture Research. Anna leads the development of the thematic review on aquatic genetic resources. Dr. Sonnesson is currently the research director of the Department of Breeding and Genetics of Nofima in Norway. Dr. Sonnesson completed her PhD in 2002 at Wageningen University in the Netherlands. Between 2002 and 2019, she worked as a senior research fellow at Nofima. Her research interests are mainly on optimization of breeding programs for major and emerging aquaculture species, with special focus on methods to include genomic information in selective breeding, managing genetic variation in breeding programs, and gene mapping. Dr. Sonnesson is a highly experienced fish geneticist. With us today also have Dr. Graham Mayer from FAO. Graham is a senior aquaculture officer with the Fisheries and Aquaculture Division of FAO. Graham is an internationally recognized leader in the aquaculture sector, specializing in the conservation, sustainable use, and development of aquatic genetic resources for food and agriculture. He is currently leading the initiatives to develop a global information system on aquatic genetic resources and the development of global plan of action for aquatic genetic resources. Prior to joining FAO, Graham spent 8 years as a program manager for production innovations with the Australian Seafood Cooperative Research Centre. Graham has also been heavily involved with the World Aquaculture Society, serving as its president during 2013 to 2015. We also have today Dr. Eric Halleman from Virginia Tech. Eric is a professor in the Department of Fish and Wildlife Sciences at Virginia Polytechnic, and also the State University in the US. There he teaches courses in general and conservation genetics. His research concerns are genetics of cultured fishes and population genetics of fishes, mollusks, and other taxa. He engages actively with fishery management agencies, the American Fishery Society, the FAO, and the many other international bodies. He is active in international efforts to coordinate oversight of animal biotechnology. Our last panel member is Dr. Fran Humphreys from the Queensland University of Technology. Dr. Humphreys is an academic, consultant, and mediator in aquatic biodiversity, biotechnology, and marine law and policy. She is a chief investigator on marine and artificial intelligence projects at the Queensland University of Technology Law School in Australia. She is on the International Council of Environmental Law delegation for the United Nations negotiations of the proposed treaty for the conservation and sustainable use of marine biodiversity in areas beyond national jurisdiction. She was previously a senior research fellow at the Griffith University and spent a decade with the Queensland government as a fisheries manager, mediator, and legal advisor for biodiversity, fisheries, and aquaculture. We have a full, very competent group of presenters and, of course, discussions during the panel. The presenters, the floor is yours. Thank you.
Thank you. Yes. Yeah, hello, everybody. It's very nice to be here. Welcome to the most interesting session of the whole conference. I would like to thank the organization first that I was invited to work on this thematic review. It's been very interesting, a lot of work, but now we are here and we hope you like it, of course. I would have preferred to meet you all in real life in Shanghai, but now we have to stay at home, and I hope to meet you some other time. And I really hope we can get a good discussion after our presentation. So my name is Anna Sonesson. I work for Nofima in Norway. I will start off with a couple of slides. So when we talk about aquatic genetic resources, we work with many species. Some are finfish, we have echinoderms, we have shellfish, seaweed, crustaceans, and aquatic macrophytes. There are 700 species reported as being cultured in the world, and all with unique biological features and all in different stages of their development. In this session, we have 4 subsections. The first one is on genetic diversity in aquaculture. The second one is the introduction of invasive species or farm types, so it's a risk assessment. The third one is on access and benefit sharing of aquaculture genetic resources and associated traditional knowledge. And the fourth one is on genetic improvement. The outline for each subtopic will be the same. First, you will hear some words about the current status, then the challenges and needs, and finally, the way forward. Now, then I would like to give the word to Graham Mayer, please.
Thank you very much, Anna, and for the introductions. My role today is to describe the part of the paper that deals with monitoring, characterization, conservation, and sustainable use of our aquatic genetic resources. Next slide, please, Anna. The current status that I will outline now comes primarily from a publication of the FAO, which is the first ever global assessment of the status of aquatic genetic resources in aquaculture. This came from 92 individual country reports, with those countries representing 96% of global aquaculture production, so it is quite representative of the global situation. This was published by FAO in 2019. Next slide, please, Anna. So just a summary of the highlights of aquatic genetic resources, which differ quite significantly from terrestrial resources used in agriculture. We farm an enormous taxonomic diversity. Anna's already said it's 700 species reported in the global assessment. However, very few of these species are thoroughly domesticated and have been developed into recognized farmed types. We have a constantly increasing diversification of species. New species are being added to our list every year, and yet a relatively few species dominate our production volumes. The top 10 species in aquaculture production represent around 50% of global production, and if we look at finfish, the top 20 species represent 85% of finfish production. So we are focused on a few species. It's important to recognize also that we still have a strong interaction between farm genetic resources and wild relative genetic resources. We sometimes harvest our seed from or broodstock from the wild, and also we introduce farm types accidentally or deliberately into the environment. Next slide, please, Anna. So talking first about conservation, what is the current conservation status of our farmed aquatic resources? First of all, the really good news is that all of our cultured species still exist in the wild. This is certainly not the case for terrestrial agriculture. So that's good news. However, the report indicated that some species are under threat. We tried to do a more in-depth analysis of the species reported in the report against those that are monitored by IUCN. There is an overlap of something like 400 species. And of these 400 species, around 14% are considered at risk up to the category of near threatened. If we do the same thing for the CITES appendices, around 4% of these species are restricted in trade. The reasons for this threat are multiple, but includes overfishing. However, 60% of these species are not assessed at all by IUCN. We know that these wild resources are essential reservoirs of diversity for aquaculture. And here I would like to diverge a little bit. Anna, next slide, please. Here we see just 4 examples of those species that are considered at risk, and they include some of our most well-known aquaculture species globally. Next slide. Thanks, Anna. So I think it's an understood paradigm in today's world that biodiversity underpins sustainable growth in food production. However, when we discuss this and when we look at our SDG targets and our CBD targets, aquatic diversity is often left out of the discussion, sometimes completely. It's beholden on us to ensure that aquatic resources are on the agenda, but the key thing here is information. If we don't have information about genetic resources, we can hardly expect them to be considered in these agenda in an active way, and certainly to be monitored and have indicators. Next slide, please. I will come back to this issue of information. But let's look now at the domestication status of farmed species. This chart on the left, published in Nature in 2007, shows that terrestrial animals and plants were domesticated 5,000 to 10,000 years ago, long before we understood the principles of genetic management. But you can see that for most aquatic species in aquaculture, they've been domesticated over the last 2 to 3 decades. As a result, the levels of genetic diversity in most farm types are quite similar to those in wild resources. This means that these farm types retain adaptive capacity. And it also enables genetic gains far superior to those possible in terrestrial species. Anna will present later on that we can expect reasonably a good selection program to generate 12 to 13% per generation improvements in quantitative traits, in commercial traits. These numbers are much lower in livestock, around 3 to 4% typically, and even lower than that in crop breeding. Thanks, Anna. One of the problems, however, is that in many of our seed supply systems, we have a lack of attention to genetic management, too much focus on quantity, not enough on genetic quality. Poor genetic management can include inadequate founder stocks, just not enough fish to start off your breeding stocks. We have small effective population sizes because many of our aquatic species are highly fecund. We don't need a lot. This can be through too few broodstock or often unbalanced sex ratios, which reduces effective population size. Many aquatic species are very easy to hybridize. So we have a lot of hybridization going on deliberately and even accidentally. These consequences of these practices is we have genetic drift, we lose alleles, we lose genetic variation, and we generate inbreeding, and we have unplanned hybrid introgression, which results in the loss of adaptive potential for the future, a declining performance if inbreeding accumulates to a certain level. We have a reduced response to selection if we start to do selective breeding, and we can lose species purity and therefore the characteristics of individual species. Next slide, please. This is just one example from our colleagues at the National Bureau of Fish Genetic Resources in India. This is a comparison of Indian major carp rohu from genetic diversity from 5 wild populations and 5 domesticated hatchery populations. We can see that now over several generations of domestication, we have lost about half of the alleles in the wild population. And we can see that a minor allele in the wild has become the major allele in the hatchery population as genetic drift. So these kind of examples of change in domestication are occurring frequently. Next slide, please. What are the issues and challenges moving forward for developing and getting more out of our genetic resources? Thanks, Anna. Firstly, I've talked about information already. We have a lack of information on the status of aquatic diversity. The global assessment is a great start, but it's a one-off snapshot. When we were doing the global assessment, we found that there was a misuse or multiple uses of terminology to describe genetic resources. All of these terms listed below there are interpreted and defined in different ways by different stakeholders in seed supply. The report, State of the World report, attempted to address this to some extent. We have introduced a new term called the farm type, which is a descriptor of the farmed aquatic organism below the level of the species. And these can be strains, varieties, hybrids, etc. And each of these terms we have also defined. So all farmed aquatic organisms should be able to be described by its species and by one or more farmed types. Under conservation, we have seen that some genetic resources are under threat in the wild. However, we have inadequate information and insufficient tools to classify the status of threat, particularly nationally rather than globally. We need to identify and target primarily in situ conservation to address these species under threat. And where we cannot do in situ conservation, we need ex situ conservation, but we have limited options there, mostly live gene banks or cryoconserved, mainly for male gametes. Thanks, Anna. For sustainable use, we have generally in many supply systems an inadequate awareness of the importance of genetic management. We lack effective tools to assess the status of genetic status of farm types. These exist and used frequently in research, but not used enough to monitor the effect of seed supply systems. And we have this ongoing development of new species, And sometimes these compete for the same resources as the development of farm types of existing aquaculture species, and we need to set the right balance there. Thanks, Anna. So what is the way forward? Firstly, information is key. The Commission on Genetic Resources, in response to the global assessment, requested FAO to develop a global information system for aquatic genetic resources. Next slide, please, Anna. We have made a good start on this through funding from the German government. We have developed a prototype system called Aquagris. This collects data from multiple sources, including our regular production statistics, the country reports from the assessment, but also we have generated a purpose-designed questionnaire to collect specific information, which can be completed by national focal points. This information is collected into a database where the data can be validated, and we have an initial data query user interface, which at the moment can generate reports such as country fact sheets, species fact sheets, and issue-specific summaries. The next development of this will generate indicators that can be used, for example, against the CBD post-2020 framework. Thanks, Anna. The other key priorities are we need to identify these key resources that are under threat in the wild and implement appropriate conservation measures. We need to find a way to mainstream genetic management in seed supply systems. We must address this before we run out of genetic variation or we lose it. To do this, we need to develop and utilize in the field cost-effective monitoring tools. It would be a game changer if we had affordable, easy-to-use tools that could monitor genetic status across seed supply systems. We need to increase and balance investment in R&D between new species and breed development. And we also need to generate enabling strategies and policies for all of these above priorities to take effect. Thank you. And at that point, I would wind up and hand over to my colleague, Eric Halleman, to talk about the risk assessment part of the paper. Thank you.
Thank you, Graham. We turn now to introduction of invasive species or farm types and the role of risk assessment and management. Next slide, please. Species farmed outside their natural range and developed farm types that are differentiated from their conspecific wild types comprise a large component of global aquaculture production. Leading examples include common carp, Atlantic salmon, Nile tilapia, white-legged shrimp, and Pacific oyster. Their production targets increased aquaculture productivity, human well-being, and economic development, but they also may pose ecological hazards to native ecosystems and genetic hazards to native aquatic genetic resources. Recognition of these potential harms raises the question, how can we promote food security while protecting aquatic genetic resources? Next slide, please. Well, we can apply the principles of risk analysis, which were developed in the context of toxicology, disease, and food safety management to meet our needs. That is, principles of risk analysis can be applied to manage genetic harms posed by distribution and production of introduced species and developed farm types. Risk analysis is a formal approach to estimate the likelihood of undesirable outcomes stemming from a given action and to manage them. Principles of risk analysis have been applied to aquaculture previously, including production of non-native species and developed farm types. FAO Fisheries and Technical Paper 519 is a book-length treatment of this topic. Next, please. So let me provide a quick overview of the risk estimation process. Risk is the likelihood of harm resulting from exposure to the hazard in this case being the invasive species or developed farm type. Risk, R, is the product of the probability of exposure, PE, and the conditional probability of harm being realized, given that exposure has occurred, P of H given E. Risk then is a decimal fraction that's the product of these two terms. Risk management can affect both terms, so risk estimation is often an iterative process. Next, please. Aquaculture goes forward within a social context, and so risk assessment might best be considered as being embedded within an interactive framework involving a range of stakeholders. Shown in the left column, they'll first be engaged in the phase 1 scoping process when the project is proposed and the scope of the risk assessment and the level of allowable risk are set. In phase 2, shown in the middle column, relevant harms and risk pathways are identified, assessment methods chosen, and quantitative or qualitative risk assessment undertaken. As appropriate, in phase 3, shown on the right, risk management measures are considered and the project is either authorized or denied. Risk communication to the broader public goes forward. Importantly, ecological and perhaps also socioeconomic indicators are monitored so that the system would prove adaptive to changing circumstances. Next slide. We must recognize that exact probabilities of harm are difficult or even impossible to determine for all possible types of harm. It may be necessary to classify levels of concern regarding likely ecological or genetic impacts posed by farm types into qualitative categories ranging from low to high. This matrix relates the likelihood of establishment of our exotic species or farm type to consequences stemming from that, and then the associated levels of concern, which may range from negligible to extreme. Next, please. All right, risk management is the design, selection, and implementation of a program of actions to minimize risk to an acceptable level. The best approach to minimize likelihood of harm being realized is to minimize exposure to the hazard using some combination of geographic location, physical confinement, reproductive confinement, and perhaps also operations management. Risk communication transmits the ongoing process and results of this risk analysis to key stakeholders, including international, regional, and national officials, aquaculturists, non-governmental organizations, academic researchers, and the general public. Next, please. So we as the aquaculture sector face a number of challenges and needs. There's need to raise the debate among fish farmers about the importance of aquatic genetic resources to the sustainability of fish production in the long term. Greater application of risk analysis is needed in the aquaculture, research, and government sectors. Now, aquatic species escape from production operations and can affect native genetic resources to a degree unlike that for terrestrial aquaculture. Hence, improving confinement of farm types at aquaculture operations is needed to promote the protection of native aquatic genetic resources and adjacent aquatic ecosystems. There are relatively few empirical case studies of application of risk analysis to issues regarding farm types that we need in order to inform our deliberations. There's a need for incorporation and implementation of risk assessment and management into aquaculture development policy. Next, please. Right, against this background, then, we offer these recommendations. There's a need to develop or strengthen national policies regulating the introduction of non-native species and protection of indigenous aquatic genetic resources. We need more risk assessment studies of farm type escapes and introgression with wild relatives, as well as synthesis of findings among such studies. We need further effort in communication of aquaculture risk management to governments and aquaculturists. While reliable confinement can be achieved in capital-intensive aquaculture systems, more effort must be directed to develop and to demonstrate cost-effective confinement systems for small aquaculture operations. FAO can play a leading role in capacity building by training responsible parties, especially in the public sector, to raise awareness of risk perception, assessment, management, and communication. FAO can present reports of case studies of successful implementation of policies promoting risk analysis in the aquaculture sector. Harmonized policies on introduction of species among countries that share water flows. Next, please. Thank you, and I'm pleased to pass the floor to colleague Fran Humphreys.
Thank you, Eric. So in this presentation, I'll give some background about access and benefit sharing. I'll explain what it means for aquaculture sectors and identify some of the challenges when the laws and the policies increasingly affect germplasm transfers. I'll end the talk by offering some ways of ensuring aquaculture sectors are not lost in this complex regulatory area. Next slide, please, Anna. So the Convention on Biological Diversity recognises sovereign rights of states over their natural resources and that they have the authority to determine access to them. Access and benefit sharing, which I'll call ABS in this talk, is the framework where a provider regulates the access, the use, or the transfer of genetic resources which are used for research, development, and commercialisation. Now, most laws target these resources for their genetic material attributes. So, for example, in most cases, it wouldn't apply to simply harvesting fish and fattening them up in pens, but it generally applies to the use of genetic material for a breeding program or biotechnology. But having said that, some countries' laws are wide enough to capture farming without breeding. Now, many national ABS laws also regulate the use of traditional knowledge associated with the genetic resources, which is controlled by Indigenous peoples and local communities, and that varies across countries. Access usually requires the prior informed consent of the provider of the biological resources or the traditional knowledge. And depending on the national law, this might require consent from the government or local community or the individual. Approvals often given through permits though, or registration processes. Now, the other component of ABS is the fair and equitable sharing of the benefits from the use of the resources or the knowledge. And benefit sharing is usually governed through contracts, and that's between the provider and the user. So this is where the provider gives monetary or non-monetary benefits to the user in return for access to the materials or the information. So this diagram shows the link between the provider and the user under that CBD sort of bilateral approach. But there are other international multilateral models like the Plant Treaty, and this is where the user doesn't share benefits back to the provider of the resources, but the benefits instead flow directly or indirectly to farmers who conserve them. Now, there's no similar multilateral model for aquatic genetic resources, so users need to negotiate permits and contracts for every transaction before being able to use the materials, and this is sometimes from multiple countries. The Nagoya Protocol, which implements ABS provisions of the CBD, explicitly recognises the special nature of agricultural biodiversity and that its distinctive features and problems need distinctive solutions. Next slide, please. So, as I said before, countries vary significantly as to the resources and the types of activities that fall within scope of these ABS measures. There are an increasing number of countries that include within scope digital sequence information, even if it's used separately from the physical materials, but that's by no means common across countries. Now, measures that accommodate the distinctive features of aquatic genetic resources are comparatively fewer than for other subsectors like plant genetic resources, but they are increasing in number. So some country measures specifically exclude domesticated genetic resources or specific GRFA subsectors, but several countries do include resources from domesticated species, although it's primarily from wild species as well. So country measures also vary in the research and development activities that may fall within scope. Most would apply to biotechnology and selective breeding, but some laws may also affect conservation and taxonomic research, and even some may affect simple grow out of species, although that's much less common. Some laws ensure that ABS measures do not affect activities of specific food production stakeholders like traditional farmers, and countries do vary in the scope of traditional knowledge that falls within ABS. So some apply broadly to ecological knowledge, while others are more narrowly focused on genetic resources. But some measures do specifically refer to food and agriculture in the definition of traditional knowledge. Next slide, please. So benefit sharing obligations as well vary between countries about whether they apply to non-commercial as well as commercial uses of the resources or the knowledge. So some measures identify options with benefits of particular relevance to aquatic genetic resources, including research directed towards food, health, and livelihood security. And now compliance measures include those that ensure aquatic genetic resources and traditional knowledge have been accessed in accordance with the prime form consent of the provider country, and that mutually agreed terms have been established if it's required by the provider country. Now, for the purposes of monitoring, some countries have checkpoints where information about the resources or the knowledge is often collected along the research and development process, such as through export offices or patent offices. So these checkpoints identify the origin of the materials and whether or not prime form consent was obtained. Some countries have aquaculture or specific sort of fisheries checkpoints, like in the Republic of Korea. Next slide, please. So as you can see, there are some examples of how countries have accommodated the distinctive features of aquatic genetic resources and traditional knowledge within existing ABS measures. So this shows that ABS does not need to be a one-size-fits-all model, but on the other hand, the ad hoc nature builds complexity for cross-border transactions, which may increasingly challenge germplasm flow. So when developing and implementing ABS measures, policymakers may wish to consider the special characteristics of aquatic genetic resources, which are discussed by my colleagues in previous presentations. So there are significant gaps, though, in research about the positive or the negative effects of measures on aquaculture. So there's a need to systematically collect this information about the origin and the uses and the movements of aquatic genetic resources, as well as digital sequence information and traditional knowledge, as well as any benefit sharing agreements that are in place. So there is also a need to forecast future global movements of aquaculture farm species, including those that incorporate genetic materials from several countries of origin. And this is because prime form consent may be required from several countries, which can add significant time delays and sometimes years for permit approvals and increased costs as well for breeding programs. Now, intellectual property, including patent law, is a separate area of law to access and benefit sharing, but there are some times where these laws intersect. So, for example, patent holders can have exclusive rights to prevent others from using their inventions that incorporate aquatic genetic resources. So this can add another layer on restrictions of movement of germplasm. But generally, there is a lack of awareness by aquaculture actors and policymakers about the implications of ABS for seed supply, for breeding and research and development. And crucially, there's very little evidence about whether access and benefit sharing has benefits for conservation of aquatic genetic resources. Next slide, please. So building stakeholder understanding of access and benefit sharing then is essential for promoting certainty for the use and exchange of these materials. Sources of information are currently ad hoc and incomplete. There are many capacity building initiatives, but the aquaculture sector would benefit from more targeted initiatives in key aquaculture countries. A central normative objective for developing access and benefit sharing measures could be to maintain affordable access to breeding material, given that such access is essential for genetic diversity and food security. Another objective is to share lessons learned from aquaculture specific benefit sharing examples. Policymakers may wish to consider the actual gene flows of aquatic genetic resources and the fact that many of them are primarily exchanged among countries of the same region. So for these sorts of cases, there could be regional or special ABS arrangements that facilitate exchange between a specific region or group of countries. And this could be under pre-agreed conditions. Now, since many ABS laws are not new, they are new, little is known about how they operate in practice. So national or local case studies on the effects of ABS will help policymakers and aquaculture actors. It will help them to understand how ABS might contribute in practice to conservation, as well as sustainable development, fairness and equity, and food security objectives. So now, next slide, please. I'll hand you back over to Anna Sonnesson for her presentation on genetic improvement and the role of public and private sectors.
Yes, thank you very much. Yes, I will talk about genetic improvement and the role of public and private sectors, dissemination and seed supply. So first, what is selective breeding? It is a method where we select broodstock that are superior for important traits. It can be one or several traits that are put in an index. And the result of selective breeding is that the performance of the entire population is improved in the next generation. So what is the impact of selective breeding? So we can use selective breeding to improve traits such as production efficiency, product quality, disease-resistant traits. We can also use selective breeding to adapt populations to a changing climate, for example, through selection for temperature or hypoxia tolerance, or to improve feed efficiency. An average of 13% of genetic gain per generation for yield has been reported in a review by global aquaculture breeding programs. In the figure to the left, you can see an example of the effects of selective breeding in Norwegian salmon industry. The outbreaks of the viral disease IPN is decreasing dramatically, with an increasing use of eggs from broodstock that have beneficial gene variants. So selective breeding can have large effects in the industry. So how does the selective breeding world look? The short answer is that each population has its own system. And one reason is, of course, that we work with species with very different biological limitations. For example, generation interval, and how much we can control the reproduction. The design of the breeding programs and the business model, they differ. Sometimes the breeding programs are specialized, and other cases, the breeding activities are incorporated in the production activities of a company. There are different ownerships. Sometimes they are owned by private companies or by governmental bodies. And finally, the technological level of each breeding program is differing a lot. For example, how much genomic information is used when performing the selection. So selective breeding is utilized in natural genetic variation. And this is very important already when you set up the base populations. They need to be carefully set up and managed. If you start off with a too narrow genetic base, you will not get any genetic gains in the later generations of the selection program. It's also important to note that selective breeding is not equal to GMO. Selective breeding is only utilizing the already existing genetic variation in the populations. And the third point here is that selective breeding results in cumulative genetic gains, which means that over some generations, you have actually improved the populations significantly. So what are the challenges and needs for the genetic improvement of aquatic genetic resources? The main challenge is the low uptake of selective breeding by certain aquaculture industry sectors. In this table, you see to the left the species with the highest production globally by volume. And to the right of the table, you see the genetic improvement status of the species. And you can see that many of these highly producing species have a low or very low genetic improvement status. This means that there is a big potential for genetic improvement to make this species even better. There are lacking long-term business models. Often breeding programs are terminated when the first financing in the first years is ending, which means that this investment is just thrown out of the window. We need better IPR and benefit sharing knowledge and applications that we just heard about. And we need an overall improved know-how at all levels, which means of selective breeding in the companies, for policymakers, etc. So what is the way forward and the recommendations we would like to give? First of all, that the acknowledgement and awareness needs to be enhanced of selective breeding at the global, regional and national levels and by producers and policymakers. We need to set up long-term financing schemes. Sometimes they can be a cooperation in between public and private entities. Like we need to utilize that some species are actually well developed, such that technology transfer from these populations can be used. And we need to increase the capacity building at all levels, and of course with the parallel R&D. Thank you. That was all for the selective breeding part, and now I would like to give an overall summary. First, there is a great potential for utilizing aquatic genetic resources more effectively to enhance global food production. However, utilization must be done responsibly with respect to management of conspecific wild populations and domesticate the resources under national jurisdictions. The wild conspecific relatives of the farm types make up a reservoir of genetic variation that can underpin the effective utilization of these resources into the future. And it's necessary to improve awareness of the importance of effective management and development of genetic resources, and develop strategies and national and international regulatory instruments to support and transform the current practices. Here, a global plan of action has been developed by FAO and will hopefully be adopted by the member states in the near future. Setting that plan into action is not a trivial task, but it will mean a paradigm shift for the future of the global aquaculture genetic resources. Then we have come to the end of these presentations. I would like to thank the authors of this thematic review, and of course, all the reviewers. We have got valuable comments and contributions by the reviewers and by members of the Co-Advisory Working Group on Aquatic Genetic Resources and Technologies. We have also received much appreciated information about the aquaculture sectors in China. You can see the list of the contributors here. Thank you very much. And finally, we would like to emphasize that the views expressed here are those of the co-authors and not of their institutions. Thank you.
Thank you very much. The presenters, 4 presenters on the genetic resources and seed supply presentation. Excellent, very comprehensive, very thoughtful review and a presentation. Now we come to the panel discussion session. Before we start the question and answers, I would like to introduce an additional panelist, Dr. Rose Basita of WorldFish. I guess Dr. Basita, yes, you are there. Rose is a global scientist in aquaculture and genetics with over 10 years of research and industry exposure and experience in Africa, Asia, as well as in Australia. Her main area of research focus and expertise is in the application of genetic technologies to improve farmed aquatic species and management of natural populations of aquatic genome resources. Currently, she leads a number of projects in several African countries on genetics, with WorldFish, and she has also been involved with USAID's Feed the Future Fish Innovation Lab projects in Zambia also. Before joining WorldFish, she worked as a research worker with the Aquaculture Genetics Group of the James Cook University and as a senior research officer at the National Agricultural Research Organization in Uganda. In this panel discussion, for our virtual participants, please type your question into the question and answer box. Inside the Zoom, you will find it at the bottom of your screen. As your facilitator, I will select questions from the question and answer box and forward to panelists for answer. For the participants joining the conference venue in Shanghai, Professor Chen will collect your questions and then read them to panel or invite participants to state their questions. Please provide your questions to Professor Chen. We may not get to answer all questions during the panel discussion, but the Secretariat will collect these questions and provide them to the authors for their consideration while finalizing the thematic reviews. Now it is the time for questions. Since Rose is just joined us and did not say anything, I'm going to throw the first question to Rose. The question is, Africa is the home to some important genetic resources that are widely cultured outside Africa. How can we ensure the benefits from these resources are shared equitably? Rose.
Thanks very much, Rohana. And yeah, good to throw me in the mix first. Yeah, I think since we don't have time, I'll go straight to answer that. And
Rose, we take around 2 to 3 minute max for an answer. Thank you.
Okay. Yeah, I think one of the ways that Africa can benefit from important improved strains that have already been developed, it's having, for example, the gift. Most of the germplasm was got from Africa, so to say. And I think Africa can directly benefit from these strains, for example, by utilizing the strains first and foremost. But of course, having listened to the rest of the presenter, this will be on condition that introductions are authorized by government and proper risk assessment procedures are followed pertaining to biosecurity and protecting aquatic genetic resources. So these need to be adhered to, and of course, farming practices to adhere to prevention of farm escapees into the wild. So I think one of the ways is utilization of the species and existing or improved management practices that promote aquatic, responsible use of aquatic genetic resources. The other way is utilizing the technologies that have been developed, for example, in the development of gift, for example, again, with WorldFish with partnership with other people. And using those technologies that have been developed, Africa can adopt those technologies to improve the indigenous species within the respective regions. And you mark that I'm sort of promoting sort of a regional approach because of the investment costs that are engaged in running a sustainable breeding program. So you look at regions like SADC or ESC in that way. So in terms of utilization, I can give an example, I think with the agreements that have been signed with Nigeria, for example, that in the pipeline to utilize the gift. Trials have also happened with Ghana initially to see how that works. I think that those are key examples that I can pick up from a WorldFish perspective. Thank you, Rohana. Over to you.
Thank you very much, Rose. Thank you. Let me now go to the live session in Shanghai. Professor Chen, do we have any questions from your live participants?
有，首先感谢4位专家的精彩报告。各位专家从不同角度、不同层次介绍和总结了水产遗传资源保护、研究和利用的现状、存在问题、面临的挑战及发展趋势，主要包括遗传资源的保护与可持续利用、外来物种和养殖品种的风险评估与管理、遗传改良，特别是选择育种技术的发展对水产养殖业的促进作用，以及水产遗传资源的获取与利益共享等4个方面。内容丰富，信息量大，这些内容对于水产遗传资源保护和利用以及水产养殖业的可持续发展具有重要的指导意义。大家知道，大力发展可持续的水产养殖业已经成为世界各国的共识，也是今后解决人类粮食问题的重要途径。
There's also an issue of protecting aquatic genetic resources and seed supply. And this is an issue that is of great importance to all of us. In China, we are constructing a specific aquatic genetic bank. This is in line with the government's policies and plans so that we may continue to develop relevant technologies to protect aquatic genetic resources. This is a very important mission for the Chinese government. In the recent years, we've witnessed many experts making headways in the protection of aquatic genetic resources. China is no exception. Chinese scientists, we have already worked on more than 50 species of crustaceans, of fish, etc., and have contributed to the protection of aquatic genetic resources. So for us, the various fish species, we've also employed selective genetic breeding. So the new technologies are helping China to catch up with the rest of the world in terms of furthering the protection of genetic resources. So the selective breeding, it is an important development in aquaculture. And we believe this is the new generation of technology that will be employed and will help us to further enhance and progress the development of aquaculture. And this, of course, will contribute to the global quality of aquaculture management. So we would like to raise 3 questions. So first question comes from the Institute of Chinese Aquaculture. So please, you may have the floor.
Thanks for speakers give us a wonderful report. As we know, gene editing is a very promising technology for aquatic species breeding. Would you like to introduce how to this technology to improve the traits such as growth performance, resistance to the disease? Thanks.
谢谢。第2个问题来自中国水产科学研究院长江水产研究所的李创举研究员，请提问。
Thanks for a good presentation. My question is how to identify effective population size efficiently in agriculture genetic breeding.
谢谢。第3个问题是来自中国水产科学研究院黄海水产研究所的周倩研究员，请提问。
Thanks for the talks. So as we know, the genomic selection technology has made great success in the aquatic breeding, and the SNP array is an important tool for the promotion and the application of the genomic selection. So is it possible for the FAO to initiate a project to construct SNP arrays for some important aquatic species? Thank you.
现场提问结束，谢谢。
Thank you. Thanks very much. Thanks, Professor Chen, and for your statement as well as the 3 questions came up. I must confess that I'm not completely clear with the 3 questions because the interpretation was not, sort of, I couldn't really get in English. But the last question was about the population size of a genetic program. The first one is leading to FAO regarding species genetics improvement. Graham, did you get the question 1?
I understood both questions. I would suggest that that Anna can address the gene editing and genomic selection, but I can address the effective population size and the question directed for FAO. So the question was, how can we know and estimate the effective population size in aquaculture breeding? This is difficult. I mean, the fundamental equation is very simple, but you need to collect the data. So I think with managing effective population sizes, there's 2 things that we can do. One is that we can issue guidelines that can raise awareness of the importance of managing effective population size. It's a fairly simple principle, and essentially it requires monitoring of the number of broodstock that are contributing to the generation of the next generation of broodstock. But I think where that's not possible, I think it's important that we develop some genetic tools where we can actually monitor the effect of what's happening in seed supply systems, and we can identify bottlenecks. So the question was also about SNP panels and whether FAO can promote this. FAO is not an R&D organization, but I think FAO would very much support the technical bodies, including World Fish, which is represented here, to support the development and dissemination of tools such as SNP panels, which can be used to monitor the genetic status within seed supply systems. These tools generally exist and are used in research, but they need to be developed specific to major aquaculture species and then made available in a cost-effective way. One possible way is that if we actually look at environmental DNA rather than sampling multiple species, multiple individuals, we can bring down the cost that way. So FAO would definitely support the development of policies and practices that make this monitoring more available. Then I would suggest to throw the question on the role of gene editing in the future of aquaculture and genomic selection to Anna. Thank you.
Yeah, okay, thank you. Yeah, so if I understood the question well, it is if we can utilize gene editing in selective breeding. Was that the question?
Yes.
Graham, yeah. The basis is selective breeding if we want to do genetic improvement, because we need to have the quantitative measurements and recordings in the population. However, if we have knowledge about single genes, then there are probably ways, we are working on that, where we can actually incorporate this information into the breeding programs. But we should then also remember that most of the traits that we work with, for example, growth, they are not steered by only single genes. These are complex traits that are steered by multiple genes. So it makes this technology less useful for those traits. But for other traits, actually there was yesterday an example in the news where they edited for a very important viral disease in Atlantic salmon. So it is of course possible to utilize gene editing there, but it has to be incorporated into the current selective breeding, because you don't do any selection if you are doing gene editing per se. So that would be my answer to that question.
Thank you very much, Anna. Thank you very much.
Can I also give a comment to the estimation of effective population size?
Please.
So we are actually doing that. We have if we have high density SNP ships, we can actually estimate at least the current effective population size quite adequately. And if we have even more SNPs and a representative sample of the population, then we can even go back in time and see how this effective population size has developed over time.
Thank you very much. Thank you, Vanna. Right, let's go to the question and answer box.
May I add to the genome editing question?
Yes, please. Go ahead, Derek.
Okay, thank you. Genome editing can contribute to aquaculture breeding. Let me agree with Anna in as much as you'd be adding this trait to a selectively bred line, or else you would be losing progress. But there are some interesting candidate gene modifications that are being posed for aquaculture production. Specifically, knocking out expression of the somatostatin gene. There have been lines developed for tilapia and for red sea bream. But before these sorts of applications can be brought to aquaculture production, you need to have three things. And one is an enabling regulatory environment that's scaled to risk. And to my knowledge, only 2 governments on Earth have really gone to the point where they have these regulatory systems in place. And those would be Brazil and Argentina. You need an infrastructure because you are dealing with a GMO. So you'd have to be able to confine these animals on the aquaculture operation. And lastly, and perhaps most importantly, you need to have a permissive consumer perception of this, which would require some serious marketing to make the point that these animals are safe to consume, that they offer value to the consumer, that they perhaps are produced near the market and hence have a smaller carbon footprint or such. And unless you have those 3 conditions, that these products, these genome edited products, will not find widespread application in aquaculture. Thanks, Rahana.
Thank you very much. Thanks, Eric. Thank you. Good questions and good discussion on the 3 questions came from Shanghai. Let me take one question from the question and answer box. This is to Fran. The question is, the harvest of wild cardinal trees from the Rio Negro in the Amazon helps conserve rainforests and communities in the area. Now these fish are being bred in the developed world, allowing for reduced price in pet stores. How does this fit in with the CBD ABS considerations? To you, Fran.
Thanks, Rohanna. And that's an excellent question because it does go to the heart of access and benefit sharing, which essentially, when you've got international transactions and genetic material is going between borders, there is this disconnect between the access and benefit sharing measures that may be in a country where the resources were collected, and then they may be used in other countries, in which case there's a different set of laws that may apply to that. So the Nagoya Protocol was trying to address this with a fairly innovative approach, which they call user compliance measures. And this is where countries are required or obliged to create measures in their countries that ensure that the prior informed consent and mutually agreed terms have been complied with from the provider country when they're used in the country where the ABS laws are. So the issue is that not many countries have user compliance measures. The European Union countries do, and there are Norway and a few other countries have these measures. But a lot of the time, it really just depends on when you're using them in other countries. It's very difficult to, I guess, ensure that those materials have been subject to prior informed consent and mutually agreed terms from the provider country. Now, in this case, I guess if they were collected from Brazil, Brazil has a different sort of approach, I guess, to access and benefit sharing now. They used to have prior informed consent for genetic material and traditional knowledge. at the time of access, but they've moved to a registration process where it's more of an end user sort of mechanism. So you don't need the permission at the time of collection, but you do have obligations when there are certain events triggered, like commercialization or sort of end use of those genetic materials. But I guess getting to the heart of this question, it's very difficult to, if they're being used in countries overseas, it's very difficult without those user compliance measures to ensure that the benefits flow back to the provider country. And in this case, there would be traditional knowledge involved, or there could be traditional knowledge involved as well. So that I guess is the crux of ABS, where it needs to be more of a collaborative approach between countries to ensure that you've closed that compliance loop.
Thank you very much, Fran. Thank you for that answer. Let me take 2 more questions from the box before I go back to the Shanghai group. This question is for Graham. Have you estimated global value of aquatic genetic resources and its change versus breeding investments? Graham.
The simple answer to that question is no, but I would very much like to. I mean, we essentially know that that the value of genetic resources is essentially similar to the value of aquaculture production. But in terms of investments into genetic development of farm types, we don't actually know what that is. And in terms of the economic values of the gains, in some cases we know. I think Anna could probably tell us the economic value of the salmon breeding program in Anna, which I think in Norway, which I think is equivalent to about 1/3 of the increase in the value of salmon production in Norway. But I think it's the potential opportunity of increasing genetic improvement beyond current levels, I think, is absolutely massive in economic terms. Currently, we estimate that we are only selectively breeding 10 to 15% of our production, so the rest of it is relatively unimproved. If we could increase the rate of uptake of selective breeding so that in a decade's time that number is 25 or 30% of production, we cannot change it overnight, it's a long-term approach, then I think that could have an absolutely massive economic value. The cost and return on breeding programs has been estimated and is generally extremely positive. So generally speaking, investments in well-managed breeding programs give excellent returns. So I think it would be nice to do some modeling to get a big picture to say we need to promote selective breeding, but if we could say, if we did it and we increased from current 10-12% to 30%, it would give an economic value. But I think it could make a major contribution to addressing the increase in supply of aquatic food without necessarily increasing the resources needed to deliver that supply. Thank you.
Thanks, Graham. Good answer. Is any other panelists would like to add 30 seconds, 1 minute into that question? All right. Thank you very much. Before I take another question from the box, Professor Chen, do you have 1 more question from your group?
没有问题了。
No, right.
No.
That's fine. Thank you very much. I will go back to the question and answers box. Right. This question is for both Eric and Rose. African aquaculture desperately needs the best tilapia and catfish for aquaculture, but is also home to the important wild genetic resources which can be contaminated by introduced farms types. How can we address these risks and benefits? Eric, you can start with, I think, and then Rose can support.
Okay, thank you. That's an excellent question. I very much appreciate it. I think Rose started answering this when we introduced her maybe 10 minutes ago, and that is to develop genetically improved farmed types appropriate to the region. She was referring briefly to the Akasombo strain, which was developed for Ghana and its region in West Africa using the GIFT technology. There's also a parallel experiment going on in Egypt using the GIFT approach to genetically improve tilapia that are sourced from the Nile Basin. The point here is the development of these regional resources, should these fish escape from confinement into natural ecosystems, would minimize the risk to the receiving populations in those regions. They would be more nearly locally adapted to the conditions in those regions. I would also suggest that my African colleagues consider genetic improvement of African catfish types. I'm not aware of any selective breeding programs for African clarius catfishes ongoing, but I'll be pleased to learn that I'm mistaken on that point. There's also a need for outreach to various aquaculture institutions on risk assessment, risk management, risk communication, so that the sorts of risk issues that I spoke about in my talk at the outset might be more effectively addressed in Africa. In terms of risk management, the important issues might be, where do you site farms? What are the appropriate species and farm types, as I just alluded to? Improvement of confinement measures that are cost effective and appropriate to the region, and then operations management measures. Okay, Rose?
Yeah, I think you've ably answered it from the perspective of using the already existing technology to improve the local types. The local indigenous species, and I think we have examples. For example, even here in Zambia, we are looking at improving the Oreochromis andersoni, the three-spotted tilapia, and the same is being used for improving Oceranus in Malawi. So that's one way of using the technology. And I think I'd also mentioned about the actual utilization of the improved strains, like the GIFT, which were already developed. But remember that the original stock was gotten from Africa, so to say. So, and I think, there were trials to compare the Akasombo with the GIFT. And that paper is out, I think it came out last year, just to show the advantage of utilizing species that have already been improved and tapping into that advantage. And that takes me to contributing to the how selective breeding can increase production. So I think looking at the other agriculture sectors, we see that in aquaculture, the gains are much higher. So looking at over 10% or 13%, the benefits on investment in breeding programs comes out faster compared to other types. You asked if there are breeding programs for catfish. I think they are starting to evaluate the possibility of improving African catfish with Nigeria as well. So yes, there are plans to start, but in order to do such a great investment, I think analysis needs to be made to see that there will be cost effectiveness of implementing such a program. Yes, there are attempts. Thank you.
Thank you, Rose. Thank you very much. Yes, very, very pertinent question, a timely question and timely answer. Let me take one more question from the box. This time, I think this question can go to Anna and Graham. Among yourselves, please decide. The question is, the contribution of aquaculture in alleviating poverty in Africa is enormous. However, to the best of my knowledge, research in molecular genetics in Africa is still very preliminary. Please, what is the state of molecular genetic research in Africa? And is there any plan to assist African countries in advanced technologies to improve their research capacity? Graham, Anna, anyone else from the panel, please.
I would suggest maybe Rose can start with that one and perhaps Anna can follow up.
Rose?
Yeah, just repeat for me. I think I missed a bit. Probably my internet jumped up a bit. Yeah, thank you.
Basically asking about molecular genetic research in Africa, and we are very sort of rudimentary, preliminary. Are there any plans to improve? Thank you.
Okay, yeah, I think that's correct. There are plans to improve, and we talked about, again, benefiting from already existing molecular resources or improvements that have happened elsewhere. And I would give again an example where World Fish, in partnership with other partners, were able to develop a SNP chip, and that was particularly for the gift tilapia. But I've used that same SNP chip now to evaluate some populations of local species here in Zambia, like macrotia as well as Andersoni. So I used it last year for hatchery types, and this year I've used it for the wild types. So yes, molecular world is moving very fast, and we need to make use of what's there because of the cost of investment. And then there are also specific resources that are being developed for African tilapia species, for example. So I would say it's a work in progress, and there's hope. We are moving somewhere. Thank you. Over to you, Anna.
Thank you, Rose. I think the answer is there. Is anything else from the panel? That's fine.
I'd like to add something there. I know of no programmatic effort to improve molecular genetic capacity in Africa. A lot of what I know of is sort of hit or miss. For example, I mean, training African students in European, North American, or perhaps Chinese or Japanese labs, but then encourage them to go back home and practice their new skills. For instance, I trained a Ghanaian student and we sent her back to Africa about 3 years ago. There's another piece to this puzzle that has to be put into place, and that is to give returning Africans the capacity, the molecular genetics laboratories they need to fully realize their potential of these new professionals back in their home countries. I know that my particular mentee is a little frustrated back in her home country. So programmatically, no, just hit or miss here or there. There's a huge potential there that can be realized.
I just had one comment there that whilst we are waiting for that capacity to develop, and it's important ultimately to have indigenous capacity, a point was made in a framework that FAO produced, which is guidelines for kind of minimum requirements for managing genetic resources. That indicated that it's not necessary actually for every country to have its own indigenous capacity, particularly for something like molecular genetics. Nowadays, this is a very international thing, and we send our samples overseas to get the results back. So just because indigenous capacity in Africa doesn't yet exist in many places, it doesn't mean that these techniques cannot still be applied to address important issues in aquaculture.
Thank you, Graham. By looking at the time, I think we have 6 minutes left. I'm going to pose one last question.
No, Rohana, sorry, Rohana, we have to finish at 5 in a couple of minutes. So I think we should wrap up now.
Sure. All right. So do you want another question or shall we wrap up?
No, we have to wrap up. We're supposed to finish 5 minutes before the half hour, 25 past.
All right. Thank you very much. I think thanks very much for the panelists and the very lively and very comprehensive discussion. And we had several questions, unanswered questions. I think the Secretariat will forward them to the authors. The authors will look at them and do any sort of a necessary revision to the documents as required. Right, Professor Chen, I think that's for you from your group, Lively in Shanghai. Thank you very much for everything, and that they have a very good statement, and 3 questions came from your group. A few more minutes, and I think everything that the documents, all you will be able to see in the conference website. And the next session in few minutes will be the stream 2, biosecurity reducing the burden of disease. If you want to stay in stream 2, you can just stay here. On the other hand, if you want to join the other streams, they have their own separate links to join. And as always, the instructions are on the website. We will take a few minutes break before we join the next session, the diseases and the burden and aquatic animal health management and disease. Shall I conclude? Thank you very much.
Thank you.
Thank you.
Thank you to all the panelists, and we will now be kicked out and the next panel will come in. Thank you.
Thanks. Bye-bye.
Bye-bye. Thank you very much, everybody. Much appreciated.
Yeah.
Thank you.